गुड मॉर्निंग स्टूडेंट्स। आज हम पढ़ेंगे जेनेटिक consequences ऑफ इन्वर्जन के बारे में। एज डिस्कस्ड इन द preceding section on cytology, among four chromatids resulting after crossing over, the two chromatids resulting from crossing over would have deficiencies and duplications. The gametes having these chromosomes will not function. Therefore, there should be considerable gametic or zygotic lethality. In plants, there will be sufficient pollen sterility. However, since the products of single crossover will not function and the only crossovers were recovered will be double crossovers, the observed frequency of recombination between any two genes in question will be considerably reduced due to this reason. Inversions are often called crossover suppressors. This reduction in crossing over is not the actual reduction in cytological crossover. Here is the diagram of chromosome pairing and the products of crossing over in pericentric inversion in heterozygotes in Pachytene configuration. And the second one is change in chromosome shape due to pericentric inversion. Here is in first diagram there is a normal chromosome, and during the inversion it makes V shape, or we can say metacentric, and in second position the centromere change their position due to inversion and it becomes submetacentric. And third one is steps leading to overlapping inversions. The result of lack recovery of the products of single crossover. This property of inversion has been utilized in the production of CLV stock used by J. Muller for the detection of sex-linked lethal mutations. Overlapping inversions. Sometimes a second inversion is induced in a chromosome which already has one inversion. This results in overlapping inversion. If the segment involved in first and second inversion contain a common region, the gene order and meiotic configuration found in an inversion heterozygote of this type. Inversion in Drosophila populations. Inversion are known to have played a significant role in evolution of different species and races of Drosophila. This knowledge is particularly available in Drosophila due to the ease of identification of inversions in salivary gland chromosomes. They also occur in plants but cannot be so easily worked out in the absence of giant chromosomes in Drosophila. However, it is obvious that inversions occurred spontaneously in nature and became established in populations due to the adaptive benefit they conferred. Due to adaptive value, these inversions are restricted to definite localities, and the different races actually derived their names after these localities. Dobzhansky and AS2 and even deduced evolutionary relationships between races. On the basis of overlapping inversions, these relationships are shown in the form of a tree figure in the diagram of 24.3. And corresponding Pachytene configuration, there is a loop in the normal chromosome and another stage pairing at Pachytene in inversion heterozygotes. And third one is pairing between normal chromosome and the chromosome with overlapping inversion. So it all occurs during the Pachytene stage of the meiosis first in plants or animals like Drosophila and others, and due to inversion there is a structural change in chromosomes. Earlier we discussed about in the chapter of structural chromosome we discussed about deficiencies, which means the deduction of any part of a chromosome, then duplication, which means there is a extra part is added in the structural chromosome, translocation, where the parts of chromosomes are translocated in their positions or vice versa or reversely arranged, and final the inversion. During the Pachytene stage, inversions are occur between the heterozygotes and they form gametes which are viable, and it causes structural changes in their gametes form. So it's finished. Thank you, students.